In a world plagued by harmful algal blooms, humans continue to add more and more nutrients to freshwater ecosystems. Though these systems have some removal capacity, the level at which humans pollute has saturated this capacity in many streams and rivers. Many have argued that freshwater will become our next limiting resource for human survival. This highlights a critical need for understanding a stream's ability to buffer nutrient loads. The stars of this showdown between excess nutrients and the stream's buffering capacity are heterotrophs and autotrophs.
Now, you might be asking yourself, what are these little-known heterotrophs and autotrophs? Heterotrophs are bacteria that acquire energy from consuming organic carbon. Autotrophs, such as plants and algae, acquire energy externally from sunlight and inorganic carbon. Both require nutrients for cell growth and maintenance. Our story takes us to the Notre Dame Linked Experimental Ecosystem Facility, or LEAF, where two graduate students are on a mission to answer the who's who of nutrient uptake. LEAF is a unique study site because it is not as controlled as a lab experiment, but not as uncontrollable as a field experiment. This makes it a good middle ground for creating realistic experiments that can shed light on big environmental problems. At LEAF, the graduate students had access to 4 streams fed by low-nutrient groundwater, all from the same source. These characteristics made it a perfect place for the graduate students to ask, what are the relative contributions of autotrophs and heterotrophs to uptake of nitrogen and phosphorus from the water column?
To separate the autotrophic and heterotrophic contributions to nutrient uptake, they shaded all 4 streams for 4 weeks to exclude autotrophs. The final 4 weeks of the experiment, the streams were unshaded to allow both autotrophs and heterotrophs to grow. Differences in nutrient uptake between the shaded and unshaded weeks allowed them to separate the autotrophic and heterotrophic contributions to nutrient uptake.
To carry out their experiment, they released a small amount of nutrients, including nitrate, ammonium, and phosphorus, into each stream and measured nutrient concentrations at 5 locations along a 50-meter reach. The change in concentration downstream gives the graduate students an indication of the demand for nutrients in a stream.
Important in their quest to understanding the role of heterotrophs and autotrophs is the size of the substrate in each stream. We know that larger substrate, like cobble, is good for biofilm colonization, while small substrate, like sand, can be like kryptonite for biofilm colonization. What is not understood is how the variability in substrate size affects the relative contributions of heterotrophs and autotrophs to nutrient uptake. This is vital because human intervention in natural ecosystems often reduces substrate complexity.
Continuing on the quest to combat the effects of humans in natural ecosystems, the work of these dedicated graduate students, as well as their trusty technicians, adds one piece to the greater puzzle that is the cycling of nutrients and the flow of energy in streams, and brings us one step closer to saving the world from harmful algal blooms.